Yeah, sure. I'm Arne, it's Anthony and Stephen, and we're going to present our paper on a surface-based deep learning tool for the design of ternary complexes. And as Hannes already mentioned before, I'm going to briefly introduce the method part, but it's actually quite an old method that we just repurposed to this new design task. So yeah, I'm going to try and keep it brief, and then Anthony and Stephen will take over to explain the new designs and how we experimentally validated them and maybe some applications. Okay, so this method that we're using is called massive, and the idea is that if you want to design protein-protein interactions, or in our case, ternary interactions, basically all you need to know about them should be available on the protein surface. So we can map all kinds of different features onto the surface. We care about the geometry and the chemistry. So for example, the curvature describes where we can find pockets or knobs on the surface. Then we have something like a hydrophobic index that we can assign to all the mesh vertices. We can find where the H-bond donors and acceptors will be, or the potential H-bond donors and acceptors, and the electrostatics. And so all these features are assigned to the surface of vertices, and then we apply some form of a geodesic convolutional neural network on top. So all these kernels are learned, and we get a learned fingerprint descriptor out. And as I mentioned before, this method is rather old. It was originally published in 2020 in this Nature Methods publication down here. So how do we train massive? There are three different ways. I'm not going to talk about the first one because it's not really relevant to this paper. The second one is what we call massive site. And here we have a data set of proteins on which we know where other proteins bind. So we can label all the vertices as binding sites or non-binding sites, and then task massive to predict where the binding sites are. The second one is slightly more complicated. So for massive search, the idea is that we have pairs of proteins, and we compute descriptors first on protein A in the standard way that I just explained. And for protein B, we first flip all the input features. So for example, positive charge becomes negative charge, and then pass it through the neural network, and we get a different embedding out, and that should be the perfectly complementary embedding. So we can take these two embeddings. Yes.
So how do you train this with some type of contrastive learning then?
Exactly, I was getting at that. So then you define points that are in contact of your complex as positive points. You also define random pairs, and then you apply contrastive learning. Does that answer the question?
Yeah.
So yeah, basically for all the rest, the idea is that you push the descriptors closer to each other if two points are in contact, meaning they should be complementary surface patches, and you push them apart if you have a random pair of descriptors. So then the distance between two descriptors should tell you how complementary locally the surfaces of two proteins are. And then in massive search, we made use of this by pre-computing protein surfaces on a big database. And then we have a query protein, and we can very rapidly screen this database by just computing descriptor distances to all these pre-computed descriptors. And we find the ones with the lowest descriptor distance. And those are highly likely complementary surface patches that like really nicely fit onto our query protein. And then basically the only thing that we did computationally in this paper differently to what was done before is that now we're also incorporating small molecules in the surface. And this is possible because Massive was trained on these nice generalized features, on these geometric and chemical features. And we can, in fact, just compute all of them also for small molecules. So basically, there was just some feature engineering done so that we map the features of the small molecule into the same input space as Massive expects that was trained on proteins. And then we can really just take the same Massive architecture that we initially trained on protein-protein interactions and apply it to these, what we call neosurfaces, like the combination of a protein and a small molecule already bound. And then with that, the idea is, let's say we have a protein like in the top left corner here that is at first predicted not to have any like clear binding site. as soon as it gets in touch with the small molecule, we see this patch lighting up in red, which means that massive predicts there to be a binding site. So this is this massive site idea that I mentioned before. But we can also do the massive search stuff then based on this input patch. We can screen our database of all kinds of proteins and try to find something that fits well to this new neosurface and dock it.
Sorry,
And then of course,
when you do massive search, do you have when you train this, how many binding sites do you have, or how many surface complementary embeddings or representations do you have per, let's say, protein-protein interaction?
you mean for one given protein-protein interaction, how many vertices are in touch? Or
no, I mean you can have around each vertex, or from each vertex, you could now get one surface representation, no? Like by looking at its
one embedding
surrounding
associated
surface.
with each vertex.
Yeah, but now, like, which do you say are the two complementary ones? The two complementary representations. So you could also take the vertex that's at the other side of the protein, right? And that would make sense.
Yes, you define the binding site first. Usually you have an idea where. Well, you can either just use massive site that predicts where the binding site is and use that as your ground truth and automate it that way. But very often you also know where you expect the binding site to be. And then you can manually define.
No. Right? In our case, we have two proteins interacting with each other in our data. So we know where the binding site is.
Yes.
But now, is this a single training pair? Or are we like, these are two huge surfaces? Now we take the surface representation here and the surface representation here. And the one here and the one here.
Yes, all of them are training pairs. All of them that
so we feed all of. Okay, so we feed every single one and pool them in some fashion, or do we take the ones that are the closest to each other and then have many training pairs?
Yeah, all the closest one basically. For each interface point, you find the closest one on the other protein, and that's your positive pair. But since, as you say, the interface might be several square angstrom in size, you will have a lot of different vertices that fulfill this criterion, and you can all use as positive examples.
Okay, nice.
All right, and then this is the last bit about the computational part. Of course, we need to benchmark this a little bit. What we did here was we took 14 ternary complexes that just people in the lab knew about or had experience with. And then we split them into two cases each, because basically you can now assume that the small molecule is either part of protein A or part of protein B. So overall, we had 28 benchmarking cases. We processed them with massive. We put the correct binders in some kind of big pool with 9,000 other decoy proteins, and then use this massive site approach to screen this pool and see if we can recover the best binders in the top hits. And this worked for about 70% of these cases. And just as a negative control, we also just removed these small molecules from the interface and saw if we recover something. And as you would expect, if the complementarity is gone because the small molecule is missing, you also don't recover much. That's the blue curve on the slide. We also wanted to compare to a co-folding method. At the time, AlphaFold 3 was not yet available, with custom ligands. So we had to resort to Rosetta Fold all atom. And with the exception of these few complexes up here that massive couldn't resolve at all, it usually has lower RMSDs than Rosetta Fold all atom. But here, of course, we need to mention that Rosetta Fold all atom is a co-folding method, while massive does rigid docking. Okay, so if there are no
number of solved complexes. I thought, are you evaluating here the search? No, like how many? My understanding is that you have a test set with like 30 or 26 complexes, and no, I don't know what's going on. The y-axis is the K in the top K.
So the K is the x-axis. So you can see that for most complexes that we find them in the top, I don't know exactly what this is, let's say 10 or so. That's why the curve is so steep at the beginning. Then we find some later on.
Is it just that you have a test set with 27 complexes or so?
Our test set is 28 complexes, yes.
28, okay. And then you just check, do you find the binding partner? If you look into the top K or into the top 10 most similar complexes as ranked by Massif. And
exactly.
That's that. But then, so that's kind of how I interpreted the left plot already, but how does this give you a structure prediction? Do you then just like I don't know, make the most shape complementary thing, or how do you do the search with RFAA if it's not about the structure prediction?
So I didn't really mention this. I didn't want to go into too much detail, but the first step is just a fingerprint search to find a complementary patch. But then the second step is a point cloud alignment that also takes into account these embeddings on all the vertices, together with the positions of the vertices, and tries to align the point cloud of the found potential binder. So this is just a rigid body docking, basically, the second stage. And from that, we have we can RMSDs and everything.
What's the purpose of that? Like, why do you actually use that in your design pipeline, this alignment?
Yes, because we rescore them. Even though these descriptors allow us to very rapidly screen the database, they're still quite noisy if you do it on a per vertex basis. But then after this alignment, we have many, many aligned vertices at the interface, and we can rescore based on all of them and reassess the complementarity. And that's also what determines the final ranking.
Yeah, you could put it in like, or you could assess the, no, you can't do that. Yeah, no, makes sense.
Okay, and then I'll hand over to Anthony. We'll talk about the design pipeline.
Yeah. All right.
Which I presume will also include these rescoring methods that you mentioned.
Yeah. So as I will mention, the pipeline is quite similar. It's just that this time, as you will see, we have a different database in order to make de novo binary design. So you usually start with a protein in complex with a ligand that you know and you want to target. And one important thing is that in that case, the ligand needs to be exposed to the solvent if you want to design a de novo binder that will recognize this protein ligand complex. Because if the ligand is deeply buried in a pocket, well, you won't be able to have a de novo binder that will sense and capture this neosurface representation. So this is one important feature that you need to know in order to design de novo ternary complexes. And the first thing you do is to ask Massif, where do you have more chance to get a novel protein interaction happening? Because sometimes these ligands are quite broad, as shown here, with BCL2 complex with venetoclax, and you see that the tip of this small molecule has more chance to be involved in a protein interaction than the other side, for instance. And once you find a good interface propensity site, you then extract the fingerprint of that patch, as Arne mentioned before. And then you try to search for a complementary fingerprint in the database. So very similarly as the protein interaction docking we mentioned before. But this time, this database is composed of small protein fragments that we called seeds. And they're called seeds because that's the starting point. That's the hotspot motif around which you will design a novel protein interaction. So you try to find a complementary fingerprint similarly as before, and then you get a protein fragment which is associated to this complementary fingerprint. So this time we have found and dock on the protein surface, on the protein ligand surface, I should say, the protein fragment that has the best complementarity to this patch of interest. And because we cannot simply work with small helical motif and peptides like this, we need to first optimize the fragment, because Massif will give you the best output it finds in the database, but it's not necessarily the most ideal binding motif ever. So to make this a small optimization, we use Rosetta, and we have a pipeline that will remove some of the clashes that you can encounter and make some relaxation. And then this small motif is grafted on a scaffold protein that we have from a database of de novo protein that we have collected from different previous papers. There are some mini proteins from the Baker lab and some other proteins. And at the end, you have your de novo protein design of interest that you want to test. And then we select more or less like 2,000 designs that will be tested in Yeast display, as I will mention in a few slides. So that's the overall computational design pipeline that will then be tested experimentally in the lab. And to test actually this approach, we choose 3 different test cases. So we had BCL2 in complex with venetoclax. So BCL2 is a pro-apoptotic protein, and venetoclax is a clinically approved small molecule used as a chemotherapy. We also have an antibody called DB3 that can bind to progesterone, which is a hormone. And finally, we have a peptide deformylase for Pseudomonas aeruginosa, so that's a bacteria. And you can see here actinonin, which is an antibiotic. And for all
now
this,
you want to design mini peptides or protein, mini proteins that bind to this and form a ternary complex.
So there are no ternary complexes for those, so they're purely de novo. So we try
so
to.
You want to design mini proteins that form a ternary. What's the purpose of that? Or why
Well,
are these useful?
that's some applications that we look at the very end.
Okay, so these are just to check.
Yeah. So there are different ways because, well, if I want to already skip to the end, basically anything you have in your cells needs to be regulated at some point, and most of the regulation involves a protein-protein interaction, or you can have phosphorylation, you can have a degradation, and so on. So at some point, you always have a small molecule that can bring two proteins together, like transcription factor and activation domain, and so on. And the fact of controlling this protein interaction can allows you to regulate and control different pathways in cells. So it can be useful for synthetic biology, for different cell-based therapies, and so on. But I will come back to that at the end.
Okay. So yeah, maybe we'll see some examples of that later. But these 3 cases now, they have no usefulness if we design the ternary complex. It's just a demonstration. Is that right?
Yes, it's right. But still, if you design a de novo ternary complexes, you can still repurpose it for something that does not involve this protein. I mean, it can be, as we will show at the end, some CAR T cell therapy. So you can repurpose that for synthetic biology. So it doesn't have to use the apoptosis that usually BCL2 is using.
Yeah, maybe we can get to that later, like in the details later. Yeah.
So for these different test cases, we first ask Massive, where do we have more chance to have a protein interaction happening? So we got moderate to quite high interface prediction that was done, depending on the protein ligand complex that we wanted to search for. And then we design about 2,000 de novo protein designs for each of these different test cases. And to do that in a very high throughput way, we use Yeast display. The way that Yeast display works is that you genetically modify your yeast with the oligopool libraries. So you have now DNA pieces that encode your protein designs. And then your yeast are genetically modified so that they express and display on their surface the protein design of interest. And then you have on the C-terminus of your protein a tag, which is an HA tag. Yes?
Okay, I'll use my hand raising in future, I think. Because if I interrupt, there's always a little bit of a delay, and that makes the interrupting a bit weird. So you just put the DNA of your design inside of the yeast DNA, and it starts expressing that stuff.
Yes, that's it. Yes. So is that all the question, or?
Yeah. Well, then also, like, why does it display it? Does yeast just display everything that is inside of it? Does it always display the whole protein, or just some snippets of the protein?
So as you may know, most of the organisms that we transform, they're transformed with the DNA plasmid, so the circular DNA plasmid, and they encode for different parts. And here we have, as you can see on the N-terminus, we have an AGA2 protein, and this has a secretion peptide that tells the yeast that once you express this protein, this fusion protein, it has to be secreted outside. And this AGA2 protein makes disulfide bond with a native protein from yeast, which is called AGA1. So then it will basically stick your design, your secreted design to the surface of the yeast.
So you don't actually you don't put just your design DNA into the yeast. You put now a chain, or what you encode now is AGA2 plus your design plus HA.
Yes.
And that's what's expressed. And
yes.
Then it always displays everything that contains the AGA2. Do you have,
exactly.
or did people get a crystal structure for a construct like this, like AGA2 plus design plus HA?
No, not that I know.
I doubt it. It's quite flexible.
Yeah, no, it's way too flexible because you have a huge glycine serine linker here, and you cannot solve that as a crystal. You can maybe find some AGA2, AGA1, I guess.
There might be some rigid systems, but not that I know.
But not with the design together, because this is way too flexible and will never be solved as a crystal structure.
And then why is the linker between AGA2 and design glycine serine and not just glycine?
So glycine is very flexible, so that's why you use it, but it's also not like super hydrophilic. So we put a serine time to time every 4 glycine, so that you still have the flexibility, but also you bring a little bit of polarity in order to be more soluble in water. So that's the trick.
That's good. How long is this linker?
This one, because usually we do repetition of glycine, serine. And I think this one is 3 repeats, if I remember correctly.
15 in total.
So 3 repeats.
Yeah.
Yep. So that should be it.
Okay.
Maybe I should answer some, because I see in the chat that there was some question before from someone. So I think it was on the previous slide.
Someone asked, what score are used to select the complementary fingerprint? But I mean, that's just the massive seed score, you know?
Yeah, well, there are different scores. There is the interface. So there is the interface propensity, but also the Euclidean distance between the fingerprints. And we also have a neural network post-alignment score. So once the alignment is done, we train actually a little neural network that can score between 0 and 1 to assess the confidence. It's a bit like the PLDTT of massive if you want to compare to AlphaFold. You want to say something else?
Okay, and like, how do you prioritize? Do you have different rounds of filtering with them to come up now with the 2000 that you express in Yeast?
I mean, there are a lot more filters than for the actual designs.
Yes. So there are different filtering steps. So if I go back before, if I can, yes, here we go. So you already have a filtering step here. So there are these different scores that I mentioned, the neural network score, the interface score, and the Euclidean distance. So we call it the descriptor distance score. So the distance between the different descriptor of the two. You can already make a selection here, and then we get multiple seed for one target. And we also have here some filtering based on the Rosetta relaxation and optimization. So we select only the seeds that make a certain number of hydrogen bonds. We also remove the one that make too many buried and satisfied polar atoms. And we also keep the one that make good shape complementarity and good computed binding energy. So DDG. And we do the same at the end once we have the final designs.
How do you compute DDG?
Basically, it's once you run Rosetta, you have this energy score function in Rosetta, and then you calculate the energy in complex versus in unbound form, and it gives you a difference of binding energy in presence versus in absence of the complex. So it's based on the enthalpy. It's based on the computed enthalpy. There is no entropy involved here, which is one of the biggest pitfalls, I have to say.
It's also not based on the enthalpy, right? You just look at a single structure. So you only have
but we usually compare it to enthalpy.
Value.
This is still based on some enthalpic state, yeah.
Yeah, but I like to actually get an enthalpy. You would have to average over conformations. That's yeah, and then as far as I know, that's not what Rosetta is doing, is it?
No, it's not what it's doing, but usually because we have this state of energy at different step, we still call it a difference of Gibbs energy. But that's why we call it delta G. But still, if you want to decompose it in enthalpy and entropy, we do not have an entropic contribution. We just have the enthalpy.
No, but that's what I'm saying, right? You don't have an enthalpy. You just have a potential energy difference.
It's an approximation. I mean, it's not like
enthalpy.
it's not purely quantitative as you will calculate it experimentally in the lab.
Yeah.
All right. So if I come to the yeast, or do you have something else to ask about the filtering?
I mean, it would be interesting to have a notion of like the numbers. How many do you first retrieve, and then how many do remain after the first filtering step, and then the next, and so on.
All right, so I do not have the numbers by half, but there is definitely a supplement table at the very end of the paper, where basically we got, because we have helical and beta sheet based yeast. So the beta sheet are a bit harder to graft on the scaffold. So usually we try to run more beta seed in that process. The helices are a bit easier to graft. So usually we have, I don't want to say something wrong, but here we have between 1,000 and 2,000 seeds. And after the optimization, we keep between 50 for the helical seed up to 200 for the beta sheet based seeds. And overall, this gives us, depending on the scaffold, this can give like up to 10 to 50,000 designs, and we select only 2,000 because a lot of them will just don't pass the filters.
Yeah.
But that's a very rough number. If you want exact numbers, I mean, this table at the very end of the paper will give you the exact numbers and the different filters that we applied.
Makes sense. Okay, then yeast display is understood, I think, or halfway understood. And then you like have this fluorescent thingy also attached to your target.
Yes, but also we come with an antibody that detects the HA, an antibody that is fluorescent. So I didn't show it here. But basically, the way we do this is to remove the truncated protein, because it happens that during the life of the cell, you get some truncated protein and they will get sticky and you just want to discard them. And these yeasts are then sorted by flow cytometry. So basically, your yeasts are now fluorescent or not. And they are going one by one in front of a laser. And this laser has different detector and different channel based on the fluorescence. And we have one channel which is on the x-axis. This is the expression, and that detects the HA tag. And we have on the y-axis the binding signal, and that detects if the target is now bound to the design. And ideally, what you want is to have the double positive. So you do have yeast that express the design but do not bind, because, well, you cannot get 100% success rate in your design process. So there will be lots of design that do not bind. And then you get a few which are expressing and binding. And, well, in the first sort, usually you do not get like a lot of binding signal because it depends on the success rate, of course. But what you do, you sort this yeast, you put the gates, and then you sort the yeast that express the good design, and you sort out in the waste the yeast that do not express the good designs. And then you do a second mode
I'm a bit confused.
Yes.
How can you have non-expressing and binding? Like if your design doesn't express, how can the target bind to it?
Because sometimes your protein is a bit sticky, or it could be that if you have a truncated protein, so if the HA tag is truncated, if half of the design is expressed, you don't have a proper folding, and then you get some hydrophobic residue, which will just stick to everything, including your detection antibody.
Then what sort of binding affinities are we talking about here? Is this then very weak binding if you have these like auxiliary binding signals from the non-folded design?
Well, you have to think that on yeast, you get multiple copies which are displayed on the surface. meaning that you get some avidity effect, meaning that if you want to attach here with one arm and you have the other arm here, if all of a sudden this arm is detached, you still have the other one to attach. And this is what we call the avidity. So you cannot really assess an affinity on yeast properly, say, because of this avidity effect. And usually the affinity that you get on yeast are like 10 times higher than what you get in reality when you do classical method, biophysical method like SPR or BLI and so on, where you get a one-to-one.
Okay. Nice, thanks.
So once we have sorted the yeast that display the good binding designs in the first sort, we have this second sort, and now you see that all of a sudden we get more binding population. And then at that stage, you can be happy and you stop here, or you can do a third sort if you want to be a bit more conservative and you want to enrich more of the binding population. And what we do, we sort the binding population again and also a little bit of the non-binding population, so that when we go for sequencing of this yeast, so we do a deep sequencing, we can then normalize how many times each design is in a binding population versus in a non-binding population. So it's just a way to make signal normalization and to see how much enrichment you get for each and that design.
Okay.
And in our case, at the end of the process, we ended up with only one design for each of the protein ligand complex. It may sound like very few to get only one binder out of 2000, but this task of designing protein ligand complexes is very hard. This is not like designing simple protein per interaction, because now you get more parameter to take into account, like the ligand, for instance. And we found, yes.
When you count valid binders, do you also like we want to be specific, right? We only want to bind if the ligand is there and not to the rest of the protein. So
yes,
there's the one binder. Sorry.
So that's also what you see here. So here on the top, you get the signal with the ligand, and down here you see without the ligand. So you have the target protein, but the ligand is not there, and you clearly see the difference on the yeast already at that stage.
Okay, but
which is ligand specific.
what I want to ask is the one successful binder. Would you have more successful binders if you call it successful? Also, if it just binds, no, independently of whether or not the ligand is there.
So there were definitely in that thing, in that screening, some design that bind independently of the presence of the drug. That happened. But for those, there were definitely ligand specific, as I just show at the bottom of the slide here.
Yeah. But you, like the success rate, if we allow any, like if we don't require specificity, then it is higher.
It could be, but in our lab, we have like a pipeline where usually we consider success everything that has passed all the boxes, where we check all the boxes, meaning that we express the design and then we try to make point mutation to see that the binding mode is correct, because there are some labs that just assume that, yeah, I've seen binding and then this is successful. But in our case, we always make sure that this is binding the way it should before considering it success.
Okay. So these point mutations, you just mutate your design at the ligand site, for example, and then make sure that once you made the mutation, it no longer binds.
Yeah, I'm talking about this different strategy in the next slide.
Okay.
So we have these three different complexes,
Nice.
de novo binder binding to ligand complex, protein ligand complex, and you see they have like different topologies and they have different binding mode and so on. And what is important, like I just mentioned, none of them were binding to the target protein alone. That's the red histogram that you see here. So that overlaps with the negative control, which is in gray at the bottom. And once you add the small molecule, you see now this blue histogram, and you see a shift in the binding signal, meaning that the binding only happens when the target protein is pre-incubated with the small molecule. And this happened for the 3 test cases. So we definitely see an inducible protein ligand interaction that happens only when the small molecule is added. And this is exactly what we wanted. And then you can actually assess the binding mode of these different designs. And for that, you can do what, like I just mentioned, point mutation. And I just put here a little zoom in on this protein ligand complex that has BCL2 and venetoclax. And you see this methionine here, which is at the core of the interface and makes some interaction with the ligand. You can, for instance, make a mutation to see if the binding modes still occur. So that's one of the strategy to see if your binding mode match your computational expectation. And this is what we did here. That's the binding signal that we got in presence of the small molecule with the native binder. And if you do this point mutation at the interface, you see now that the binding is completely abrogated and overlaps with the negative control. And usually the way we do it is that we take like an hydrophobic amino acid that you have at the center of the interface, and then we put a very bulky and charged arginine so that it should, in principle, abrogate the binding if your binding mode is correctly predicted. And another way that you can assess that is to use a drug analog so that you still target the same site, you don't mutate anything, but now you change the ligand that bind to the same site. As you can see here, this is in blue, the binding mode that we got with venetoclax, and if we use this analog, then we do not see any binding anymore, meaning that we are also ligand specific. And the last way, which is one of the most elegant in my opinion, this is to do a site saturation mutagenesis. And to do that, you mutate all amino acid position to all possible 20 amino acid one at a time. And then you redo an oligopole library, you retransform yeast, and you do the same process as before, but this time you will have yeast that display exactly the same design, but with point mutation, so that now you can assess the effect of each amino acid at each position of the protein. And this way you can now detect the region which are more important for binding. You see now in red, that's the mutation which are more present in the non-binding population, and in blue, the one that are more present in the binding population. And you can make the average per residue position, and then you display it on your prediction, the computational prediction, and you see in red that the predicting interface is highly sensitive to mutation. So we have not done a crystal structure, but still with that, we can assess that the binding mode that we're predicting on the computer is most probably the one that we also get in reality. And also one important measurement, like we just mentioned before, this is also the affinity. So now if we predicted that they bind correctly, we also want to know how strong do they bind with each other. There are different methods where you make a titration of the ligand and you can measure it. You can measure the binding using surface plasmon resonance or biolayer interferometry. And you see here the affinity in presence versus absence of the ligand. The first version of this design show affinity between 4 macromolar, a bit more than 10, and even half a macromolar. So that's in the range of the protein interaction. That's not very strong, but still you do see protein-protein interaction that happen in the range of macromolar. So this is still good considering that these are purely de novo computational designs. But thanks to the site saturation mutagenesis data that I mentioned in the last slide, so by using this data that you see here, you see now that there are some blue spots that suggest that this mutation at this position may considerably and significantly improve the affinity. And what we have observed is that most of the time, these are like some hydrogen bonds that were properly resolved, or there was a little clash here with this serine. So if you mutate it to glycine, you improve the affinity, or this methionine that was here before was too flexible and gets too much entropy. And if you put something that is a little bit more rigid, like a leucine, you get a better binding affinity. So these are just a couple of examples, but just by using these two mutations in each target complex, we increase the affinity from 100 to 1,000 fold. So for instance, the most impressive one is maybe the progesterone binding system that switched for more than 10 macromolar to 18 nanomolar just by doing 2 single point mutation on the protein interface. So sometimes, even if you do computational design, you can be very close from an ideal solution, but only 2 mutations may matter in that case. And I think I will also tell you that the most important thing for any protein designer, these are like the structural data. So you want to be sure that what you get in reality is really what you predicted on the computer. And to do that, you can either do a cryo-electron microscopy or a crystallography. So in the case of the progesterone binding system, we were quite close, but still there was a little shift of the protein. So maybe this can be due to the fact that we are targeting an antibody that has some loops, some binding loops, and these are highly flexible, and this is also hard to predict. But in the case of the actinonin binding system, we are very close in reality to the computational prediction that we initially did on the computer. And of note, we didn't solve anything for the BCL2 venetoclax system. I guess that's part of the game. Sometimes you succeed, sometimes you fail. And in that case, we didn't get any crystal structure for that specific complex. But overall, with the mutational data that I showed before and using this different structural data that I'm showing here, we're quite confident that the binders that we predicted on the computer are really binding this way in the experimental data. And using this crystal structure, you can then also learn from your mistake and you can improve them. And I think now Steven will tell you a bit more about that, how we can improve the pipeline.
Switch seats.
Before we move on to that, maybe if you do this yeast display, right, you still need to somehow synthesize all of your DNAs, and the DNAs can be completely different. It's not just mutations that are different in them. And then if you have 2,000, how much does that cost?
2,000 design cost about $2,500.
Okay, that's pretty good.
And you're limited to 300 nucleotides.
Yes. I think now you can go even up to 400, but this is significantly more expensive.
Yeah, it gets more expensive.
So it's like 110 residues.
Yeah, ish. But also here we are, because we need a little bit of flanking regions in order to anneal to our plasmid. So we need to sacrifice a few amino acids so that we get a bit more DNA to make the molecular biology to cut and paste our fragment into the plasmid.
Mm-hmm. How big is the protein that we're looking at right now? How many residues?
This one was 60-ish, 50, 60.
60. Okay. Okay, nice.
Okay, so
Wait,
in this slide,
why is there a size limitation? Why, why 140? Why 400 nucleotides?
Well, that's the DNA provider limitation. They usually work with a synthesizer, and I think they lose a lot of confidence after a certain length, and then you can get lots of mistakes.
Okay, makes sense. Okay.
yeah, so I guess what we want to show on this slide is that if you compare the original model to the crystal structure, it mostly aligns very well, except for a tyrosine at the N terminal, which is slightly misplaced. But if you run this model through AlphaFold, it basically nails the prediction. So it gets it right. And so we just want to show that AlphaFold can and probably should be integrated into the pipeline. And then here is so how some of the failed designs have been rescued. So here you see the PLDTT from AlphaFold and the RMSD to the original design from the pipeline. And if you see the black dots specifically on the top right, so these are designs that were validated post-optimization. So maybe if I show this, so all the designs that came out of the pipeline were put through ligand MPNN, which is very similar to protein MPNN, but can incorporate non-protein components, such as these small molecule ligands. So if you redesign the amino acid sequences with ligand MPNN and then run them through AlphaFold, now you get a general shift of higher PLDTTs and lower RMSDs. So you can see that these black dots that would not have passed the filter from the original pipeline now pass the filter. And so these were tested in kind of a second round to see how much we could kind of improve the pipeline. And we got quite a big improvement, specifically for PDF1. So I think we have, yeah, here we go. So we got 12, and also these designs, instead of 2,000, it was now 500 designs per target. So if you look at PDF1, for example, instead of one design that worked out of 2,000, now you're getting 12 out of 500. So we weren't able to get one for the DB3 progesterone target, but we were able to get one for a BCL2 with this new pipeline. So it seems to give a higher success rate, which makes sense when you integrate kind of more rigid filtering based on like AlphaFold.
Okay. So do I get it right? You just run AlphaFold on the monomer that you designed, look at the PLDDT, average across all residues, not just the interface or something, and then score based on that.
Yeah. So the RMSD
And
is aligned to the original design.
okay, so you score with the RMSD.
So it was both. So it was
Okay, my bad.
PLDDT, where I think the cutoff was 87.
87,
I think it was placed at 87, and then an RMSD of 1. So basically, if after the design pipeline, if you ran it through AlphaFold and the PLDDT was above 87 and the RMSD to the original design was less than 1, then it passed this filter.
How did you come up with 87?
Well, this is well usually where you consider to get very high confidence PLDDT.
Okay.
I mean, this was also based on if you look at the orange dot up there, these are the validated binder that we showed before. And assuming that this one was validated and we also get a crystal structure from it, well, we could assume that still a PLDDT of 87 was acceptable to consider it as successful.
So you can trust it, kind of.
Yeah. Below, you're not sure, but still at 87, we managed to get a validated binder.
Okay. And it's single sequence AlphaFold.
Yes, single sequence.
Okay.
Which is even more impressive
Sounds
because the prediction it was done before with the de novo design we got was absolutely not in the training set of AlphaFold, and still it predicted completely correctly.
Yeah.
Okay. So we'll move on here. And so these are kind of some functional applications of these CIDs, as you were asking about before. So for example, the DB3 progesterone CID, it was validated in a functional assay in a cell-free system. So basically, like it's a cell-free system. So it basically just has the machinery you need to produce protein. And you fuse your interacting proteins so one protein will be fused to a DNA binding domain, such as the zinc finger, and then another protein is fused to a RNA polymerase. And the RNA polymerase by itself won't transcribe the DNA, but the zinc finger will bind the DNA. So then if you add your small molecule that induces the protein-protein interaction, it recruits the polymerase to the DNA and will bind to the promoter and start transcription. And the gene that I was transcribing here is M-cherry, which is just a fluorescent protein. So then you measure fluorescence. And then another way we validated this was in a synthetic receptor platform. So these are called GEMS, or generalized extracellular molecular sensing. That's the name of the platform that was developed by a former postdoc in our lab, Leo Scheller. So basically what it is you take your interacting proteins and you incorporate them into the extracellular receptor domain. And these receptors, so they're on two different chains, and the receptors shouldn't interact unless the small molecule is present. So you basically have each of your interacting proteins on separate chains. Nothing happens. When you give the small molecule that induces the protein-protein interaction, the chains come together, and this causes a conformational change that will eventually induce transcription and translation of a reporter protein. So in this case, it's luciferase readout. And then this was, so the previous one was extracellular. This is intracellular. So basically, you can split a luciferase into two components. Like neither of them are active. And well, one's called a large bit and one's called a small bit. And the small bit is basically just a beta strand, but the large bit needs that beta strand to be an active luciferase. So you fuse each of your interacting proteins to parts of this split luciferase. The split luciferase has a very low affinity to just randomly reintegrate. So you won't get any luciferase activity randomly. But if you have two proteins that interact yep.
So engineering something like on the top left sounds very hard, but then something like the top right or the bottom left sounds quite plausible, right? You could just attach all this other rest to your all this other rests. Maybe the top right is also very hard, but in the bottom left, as far as I understand, it's just about bringing the stuff together and making sure it stays together. And then you could just attach this gray strand to your designs, and that then achieves the deal.
Yeah, so this is a pretty like well-known assay that people have been doing for a while. But the thing is, like some proteins are better at interacting in an extracellular way just because it's an oxidized environment. And PDF1, I think, has 3 cysteines. And we tried to do it in the format on the top right, but it didn't work. So also sometimes just the orientation that the proteins have to be in each format kind of dictates what assay you can actually do with them to, make them functional.
Yeah, I mean, not that these things exist or that we want them or so. Just I'm thinking about which, like these things exist and they're useful. And then could we design them? And then presumably there are other cases where designing something similar would be useful. And yeah, for the bottom one, it seems the most plausible that we could design it.
Yeah, if you want to screen like an inducible interaction, a split luciferase system is, it's a good approach.
That's not quite what I mean, but anyway, like I mean binding, right? You have a method for engineering ternary complexes, and basically what are now the applications or the real world applications of ternary complexes where just your method, if it worked perfectly, would already enable like rediscovering this real world application. And for the top two, this is not the case, right? If your method worked perfectly, then we still couldn't solve the top two examples.
Yeah, so I think that's a good transition because the next thing we're going to talk about are CAR T cells, which the next slide is about. So basically, like what are CAR T cells? So if somebody has cancer, they can kind of analyze the cancer cells and the healthy cells, and they profile them, and they try to find biomarkers that are specific to the cancer cell, and ideally that are on the receptor of the cancer cells. You then take the patient's T cells and engineer them to target this cancer-specific biomarker. Then you expand these T cells and put them back into the patient. And basically what this allows is for these engineered T cells to specifically kill cells that have this cancer biomarker. And what we can allow is to control the activation of a CAR T cell. So CAR T cells are like a very promising therapeutic, but sometimes they're too active or not active enough, or the target that the CAR, so CAR stands for chimeric antigen receptor, is not only on the cancer cells. Sometimes they're also located. Yeah.
Guys, I may have to. Oh yeah, I have to leave at the mark of the hour. So if it's fine with you, I would leave you with the others, and or do you think we can finish in 2 minutes?
Yeah, this is the last slide. I can go through it quick.
Okay,
Here's the last
then
slide.
let's do that.
So basically, this is the format of a split CAR T cell. So if you see on the left here, this is like a second generation CAR, where right here you have an anti-HER2, which is a kind of a well-known cancer biomarker, and then you have intracellular signaling domains. So what we did, or Madalena in our lab did, is you split the activation domain from the receptor. So even if your CAR T cell binds the biomarker, it doesn't activate a signal unless the other chain comes together, which in this format is controlled by the BCL2 venetoclax CID. And some data that Madalena generated here is, so a 2G CAR is always on, and then the split CAR is, well, much more active when venetoclax is present. There's some basal activity, but you see that there's a large increase once you give venetoclax. And this can be useful because, like HER2, for example, it's on some cancers, but it's also on some healthy cells. So, yeah.
Okay, then thanks a lot from my side. Thanks for explaining the yeast display stuff and so on. That's very helpful for me. And I'm sure some others might still have questions. So if you want to stay and chat with them, that's nice. And maybe Peter, for example, has some questions. But yeah, I need to chat with Regina now. Yeah, hope that's fine.
Yeah, it's basically done now.
Okay. Then, oh yeah, we can also call it a day now. And yeah, have some finish it up here. But yeah, if some others have questions, then of course feel free to ask them. And I hope you can manage yourselves and say bye to the people once you want to say bye. Right, then bye from my side. See you.
Bye.
Bye.
See ya.
So you want to finish up that slide, or?
Yeah, so I guess I kind of said it, but kind of rushed through it. But I guess the takeaway is that in this split CAR system, you decouple the CAR T cell's ability to bind the receptor to its ability to activate a signal that will then, activate the CAR T cell to kill the cell with that signal. And this is useful for if maybe your cancer has this biomarker, but maybe some other cells in your body also have this biomarker. So if you have a constitutively active CAR T cell, this can be toxic for the patient. And if you're able to only activate it in an inducible manner with a small molecule, you have much more control over the therapy. Yeah.
All right, so we had a very brief conclusion to give you. So basically in this work, if I have to sum up everything, we provided this new pipeline that's use fingerprints and the molecular surface representation. So this time we do not really care about all the atoms. We just care about what is on the surface and what is exposed. And that's what we call the fingerprint. And previously we just consider amino acid in the massive pipeline, and we were not considering the non-amino acid components, but this time we incorporated the small molecule. And what is actually good by using this molecular surface approach and using geometric and chemical features is that you can easily generalize to everything. You don't care about amino acid or small molecule, you just care about the hydrophobicity, the proton donors and acceptor, the electrostatic, etc. So actually this generalization process is quite uncommon in deep learning because most of the time you need to make a retraining every single time. But here, because we have this higher level of abstraction, we could actually use the pipeline as it is. And with this new pipeline, we managed to design novel protein-ligand interaction. So protein that come together only if a small molecule is present. And as we just showed in the last slide, this can actually find some application in terms of synthetic biology so that you can activate your cells upon demand. This is also useful for cell-based therapies like CAR T cell in cancer. You can also imagine some biosensors. So we have this progesterone system. So you can imagine like a test that can detect progesterone or some other small molecules. So there are different applications that you can find by controlling this protein interaction with the small molecule. And with that, our presentation comes to an end. So we're still happy to answer a few questions. I've just seen that there was a question from Hannah before saying that there was a color that was not really clear. Unfortunately, we have not seen that message before. So if Hannah can just explain what color she was referring to. And in the meantime, I'm just reading Maxwell's question. Curious if in training you consider data augmenting using residue or sidechain at a distance within the monomer or only interacting proteins?
It was only interacting proteins. So the whole training set is it's just PPIs.
Yeah, so that's non-protein-protein interaction, and we just take whatever is in the distance of a few angstroms, like
maybe we can also mention here that Massive, compared to other deep learning architectures these days, is a rather small network because it's also based on these already quite informative input features. So actually with this rather small data set, that was already good enough to train the network.
All right. Does anyone want to ask a question?
Yeah,
Yes.
I had a query regarding your code. Like, the libraries that you have used are quite old. So I was like thinking, like if why not the any recent versions of the libraries like TensorFlow and others, and like is there a specific reason like with AlphaFold and something like that?
So you mean for the training of Massive? Because for the training of Massive, this is true. We didn't take new libraries, but most of the time it didn't like increase that much compared to the previous ones.
I mean, the reason is really just that we wanted to use the pre-trained Massive architecture.
Okay.
Yeah, the idea being to see how far we can push it, and if we can design these ternary interactions with it. So it's really the old model from 2020. There's nothing changed about it, and that's also why the software versions are older.
Oh, okay.
All right. No other question?
Not a question, but just really appreciate the presentation. I'm actually on the West Coast, so I've been dropping my kids at school, and but just don't normally see such a kind of applications focus here, and that was really interesting. Thank you.
Thanks.
Thank you for joining. Best regards to the West Coast. All right, so we still had a question from Hannah. I don't know if Hannah is still there, if you want to clarify. Maybe she left. But if you see any other questions that you don't want to share now, you can still send us an email. So just go to the website of our lab. Just feel free to send an email or, oh yeah, it was about a diagram that's not presented in a paper. Normally all these data are at some point in the paper. So
potentially in the supplement.
Maybe it's in the supplement.
Extended figures.
Yeah. So I don't know if you remember where it was exactly. So I don't know which one you referred to the color, because this was, yeah. Do you remember exactly where it was? because this is in a paper. This is half of it is in a paper.
About like this one?
Yeah. 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 Feel free to come off mute. That's right. Don't be shy about ligand. Yeah. Well, many slides are about ligands. Yeah. I'm not sure. Well, worst case, Anna, you can still send us a message if there is something you want to clarify. We'll be happy to answer if you just look through the
presentation.
Oh yeah, here. Okay, perfect.
I think it's still before this slide. There is a ligand target, and you were talking about selection. I think
Stuff. Was it here?
it's like point cloud.
Point clouds.
There are several diagrams on the top right
this one.
of the. Yeah, this one.
Okay. Oh, you mean the color of this one?
Yeah.
The color of the scatter plot. Yeah, they're just that they're like many points that are the same place. So this is just some density. So the more red, the more points are overlapping each other at the same position.
So it's a density graph.
Yeah, so basically the more red it is, the more dots you have that are like superimposing each other at the same place, because here we're screening millions of yeast. And if you want to see how dense this is a bit like looking at the mountain from the top, basically.
And so each dot is a cell, or
each dot is a yeast. Yeah, each dot is a yeast. So here you
yeast.
see a few dots. So there are like maybe 20, 30 yeast in that gate here. But here there are like billions of it. In each of these
And so
yeast,
why are some parts of the diagram that the density is really high, and it's like they're aggregating at the point, or it's the expression is high?
It's the lack of expression.
Yeah, here this one is the lack of expression. So that's the one, because you know not all the yeasts are alive, or they just don't want to express the protein. So you know, they are princesses. Sometimes they just do whatever they want. And it happens that here, for instance, this yeast do not express your protein, and here they do express it. And once you add the ligand with the protein target, you now see some yeast that start to bind. And then if you do a second sort, if you enrich this, you put that in culture and you try again, then now you see more yeast that express the design that bind to your target of interest. So each dot is indeed a yeast that then you sort in a certain direction. So the way it works in flow cytometry, you have a laser, they pass in front of a laser. So they're encapsulated in a little droplet. And in this droplet, you have a little bit of salt. And then you have magnet that will give a direction. So either you put them in a waste or you put them in a tube where you then put them in culture so that you enrich the good yeast.
And so the diagram is actually you take a picture of it, or it's under the microscope?
So it's laser signals. So basically on a flow cytometer, you have your yeast that go through a microfluidic channel, and they go one by one in front of a laser, and then they get fluorescent either on the green or the yellow color, so depending on the axis. And what you see on the axis here is basically the intensity of the fluorescence that the laser is measuring. So the more you are on that part of the graph, the more fluorescence you get, either on the green or on the yellow. And then it reports two values, so one fluorescence on the green, one on the yellow, and then you just report it on this X and Y axis.
So it's like it was like a flow, and then you try to how to say just aggregate all the results and then plot it like this.
Yeah, so yeah, basically that's because you have your yeast going one by one, and the more you wait, the more data you acquire, and then they start to sum up like this on this scatter plot.
Ah, okay.
That's what you
Yeah,
see here.
interesting
So
to
it
know.
takes like, so
Thank
to
you.
analyze 1 million of yeast, it takes like about 10 minutes.
Yeah, so it does like thousands per second.
Yeah.
So it's pretty quick.
Does that answer your question?
Yeah, thank you.
All right. Does anyone else have a question, yeast related or not yeast related? No question. Okay. Well, so thank you everyone for joining. And once again, if you have any questions, just feel free to go to the website of the lab and you can find our emails, or you just go on the GitHub and we will be happy to answer some of the questions that you may have a bit later. And feel free to look again at the paper and at the supplementary, because there are like many things that we could not mention in the paper for size constraints, but there are a lot of good piece of data that you may be interested in. And with that,
Thanks so
again,
much.
thank you very much.
Thank you.